So, hello everybody to our today's webinar, Oxygen, pH, CO2 in Sediments, Interfaces and Biofilms. My name is Gernot John and I'm Director Marketing and Innovation at VisiSens Precision Sensing. Today with me is Robert J. Meier. Hi Robert.
Hello everybody.
Robert studied chemistry at the University of Regensburg, and he received his doctoral degree in chemistry in 2007, and he has more than 8 years of experience in the field of optical sensors and imaging, and he's working in our department for imaging solutions. Today he will talk about the VisiSens system and how this can be applied to imaging situations in several different applications. The webinar is structured in the way we start with some general introduction and then I hand over to Robert. At any time of the webinar, please use the functionality to ask questions and we will answer them either during the webinar or afterwards in the session. If you have further questions after the webinar, don't hesitate to send us emails. Okay, we could start with a few words about VisiSens, about our company. So VisiSens was founded in 1997 as a spin-off of the local University of Regensburg, and the company is privately owned. Currently, we are more than 19 employees, and we are focusing on monitoring devices for measurement of essential parameters like pH, oxygen, and CO2. The company is certified according ISO 9001 and ISO 13485, which is the standard for medical products. Our product range covers these parameters, and we have sensors that measure through the wall, sensor spots that can be integrated in nearly every transparent vessel like bottles or shake flasks or microplates, and we have microsensors where we small microinvasive sensor on probes for measurement in small entities like small animals or also in small bottles like you see in that picture. Okay, but now we start with the presentation, and Robert, the stage is yours.
Everybody to our today's webinar. During the next 30 minutes, I want to present you some experiments on how you can image oxygen, pH, and CO2, for example, in sediment experiments, in interfaces, and also in biofilms with our two-dimensional visualization tool, VisiSens. So first I want to start on how we can measure oxygen, pH, and carbon dioxide. We in our company do it with luminescence optical sensors. These luminescence optical sensors measure via light. And they have a big benefit because they can measure non-invasively through the vessel, through the wall of your vessel. As light can transmit transparent walls, we can also detect the signals that are located behind transparent walls. Thereby, you do not need to open or you do not need to do sampling in your experiment. Thereby, you can avoid contaminations or also changing of your sample during the experiment. The sensors are not made out of glass, so there's no risk of breaking. And another very important factor is that they do not consume analyte while measuring. And this allows you also to measure at high flow velocities, at slow velocities, but also at static conditions without any flow. And the sensors are non-toxic to your sample. So today's webinar is focused on our imaging technology. Imaging in our words means making a picture of a sensor foil and thereby generating thousands of measurement points within that single picture. Thereby you can generate full analyte maps across your sample. You can compare different regions and see what's going on where in your sample. You can detect, for example, hotspots and see where most the action takes place. You can visualize gradients, for example, over interfaces. You can also monitor overtime changes in time series experiments. You can study microenvironments, visualize reactions, and do the readout in a non-contact way. And you can do all that with our VisiSens imaging systems. The measurement principle of the VisiSens imaging system is very easy and straightforward. You have a luminescent sensor foil, which is placed onto a sample, and then you use the handheld detector to excite the sensor foils and record the luminescent signals from the sensor foil. So you take a picture of the sensor foil using the VisiSens detector, and thereby you get the analyte map across your sample. You can on the one hand measure directly, which means that you can place the sensor foil directly on your sample and take a picture with the detector. Or you can also measure through transparent walls by placing the sensor foil inside behind a transparent wall and then recording the signals through that wall with the detector unit. The VisiSens system consists out of three major components. First, the hardware, then luminescent sensor foils, and the software for recording images and also evaluating the data. The hardware in here, you can see this is a small USB-powered microscope which can be plugged into a laptop or a computer, which is portable, and you can use it even in the field, or you can use it also in your lab experiments. The sensor foils are thin plastic sheets with immobilized indicators in here, which are recorded with the detectors. We have a total of 3 different VisiSens systems. They are color-coded to find the right detector and the right sensor foil for your application. The blue color code is for oxygen imaging, the red color code for pH imaging, and the green color code is for CO2 imaging. The measurement range for oxygen ranges from 0 to 100% air saturation, which equals 0 to 20.9% pure oxygen. In case of pH, we have 2 different sensor foils, one for the standard range from 5.5 to 7.5, and one for the low pH range from 2.5 to 4.5. In case of CO2, we also have 2 different ranges, one covering the low range from 0 to 1% CO2, and one covering the range from 1 to 25%. The response time of oxygen and pH sensor foils is below 30 seconds, and in case of CO2, it's below 3 minutes. The sensor foil can have different sizes with a maximum up to 40 times 40 square millimeters, which is also the maximum field of view of the detector. But it has a focus adjusted field of view, so you can also choose a smaller field of view. The number of sensing points you generate within one recorded image is more than 300,000. They are recorded at the same time in one image. And they work in a temperature range from 5 degrees up to 45 degrees centigrade.
Robert, maybe I may interrupt you. We have already a few questions concerning the device. So one is about the minimum size that is necessary. You're mentioning that there's a maximum size, but is there also a minimum size you need for the system to work?
Yes, the minimum size is approximately 1 times 1 square millimeters due to the statistical reasons and for easier handling.
Okay, that's great. That's small. And the other question is, are these sensor foils designed for reuse?
Actually, they are generally designed as disposables, but for example, the oxygen sensor foils can be cleaned with ethanol and reused after recalibration.
Okay, and then there was a question which you kind of already answered about what is the resolution of the device? What is the maximum spatial resolution of the device?
The maximum spatial resolution gets down to 100 microns, and in case of oxygen sensor foil, it can also be 25 microns.
Okay, great. I think that's it for the moment.
Okay, now let's head on. The sensor foils, in case of oxygen sensing, are compatible with aqueous solutions and also with a certain extent of ethanol and methanol, and are stable from pH 2 to pH 10. The pH sensor foils are stable from pH 2 to pH 9, and in case of CO2, stable from pH 4 to pH 9. So now let's take a closer look at the software. Within the software, you can record single images. You can also record automated time series with periods from seconds up to months. You can do pseudo-color representations and calculating and analyzing of the analyte values you recorded in your map. You can do multiplots to, for example, compare different images recorded in a time series and see what's going on in your sample. You can do one-on-one image comparison to have a closer look, for example, if you want to see the starting point of your experiment and compare it with the end point of your experiment. You can also do alpha blending to see underlying structures. And you can do statistics with the program. You can define certain ROIs everywhere in the image, which gives you an average pH value in that area and also a standard deviation and minimum and maximum values. And you can do time series profiles of defined region of interests that you place anywhere in your image and get the time plot at that specific position. And even there are way more functions within the software. So now let's head on to the practical examples. That's what everybody is waiting for. During the next 20 minutes, I want to show you how you can image the analytes in sediment samples, at interfaces, and also in biofilms. If you want to have further details on the experiments that I present to you today, I can refer you to the application notes that you can find on our website, www.presence.de. There's an application note section in the reference section. Okay, now let's head on to our first example. The first example is imaging of a sediment-water interface layer. In this case, an intertidal sediment sample was taken with a Cora that you can see here in the top left picture. And at the water sediment interface, the VisiSens sensor foil was placed inside the Cora in contact with the sample, and then imaged from the outside. In this first experiment, the pH value or the change of the pH value over time was investigated. Here on the left side, you can see a pretty even pH distribution over the sediment and the water surface on the top. This white line in here indicates the sediment-water interface, and the scale bar is around 5 millimeters. So now in this experiment, this intertidal sediment sample was overlaid with weathered terrestrial sediment in approximately a thickness of 1 to 2 millimeters. And what happens is that you can see in here in this time images, you see that where the sediment was deposited on top, the pH values drop down. This gets more visible if you take a look at the video of that experiment. So at first you see a pretty uniform pH distribution, and then when the sediment was topped with the terrestrial sediment, you can see the drop of the pH value. And this is a pretty fast change. And what now happens over time is that you can see here on the right side what happens over time. If you take an ROI of the top level where the terrestrial sediment was placed, you see a slow increase of the pH level again with time. And this is first due to the reworking of the terrestrial sediment by the microbia, which is in the original sediment, and also by proton diffusion. If you take a region of interest in the lower part of the original sediment, you can see that from its original value of 7.4, it slowly decreases due to proton diffusion.
Excuse me, maybe I can just ask about the time series, because this came up. What is the maximum length of such a time series? How long can you record it?
Actually, you can choose your interval from seconds to days to weeks and also months. You can record pictures till your hard drive is full.
Oh, wow, okay.
Yeah. So,
thank you.
Okay. Now let's head on to a little bit more sophisticated interface. This was again an intertidal sediment sample, and it was investigated in a flume experiment. And in this case, the oxygenation over the interface was investigated, And in this sediment sample, there were small burrows which were caused by polychaetes. Polychaetes are small little worms who make these burrows into the sediment for allowing highly oxygenated pore water from the water zone to diffuse into deeper parts of the sediment, and so they can breathe. So now if we take a look at this scenery in the oxygen image, you can clearly see the higher oxygenation levels in the area of the burrows, but also in the microenvironment of the burrows. And here you see a first very big advantage of 2D measurements compared to, for example, 1D profiling. If you do just a 1D profile from the top water layer into the sediment, you can have very steep concentration gradients, but also if you just move some millimeters on the next side, your concentration gradient can look totally different. Or if you match one of the burrows, you get a pretty large oxygenation. So now in this experiment, the intertidal sediment sample was again overlaid with wetter terrestrial sediment, and this closed these burrows, and thereby you get a pretty uniform oxygen gradient over the sediment-water interface. During the next 10 to 20 or 30 minutes, the polychaetes try to rework these burrows and allowing again pore water to diffuse into the tunnels and going and causing an oxygenation in deeper parts of the sediment. So now if you take a look at the time series video of that reaction, you can see in here the burrow with a high oxygenation, and then the oxygenation diminishes in here due to the overlaying of the terrestrial sediment, and then the worms reopen the burrows and oxygen can diffuse again into deeper parts of the sediment. So now let's head on to our next experiment, which is also a sediment experiment that I want to present to you. And this experiment was conducted due to this large oil spill accident happening in 2011, the Deepwater Horizon catastrophe. During that accident, huge amounts of hydrocarbons or tar balls and oily substances were spread over the sediments in the Mexican Gulf. And these tar balls can be reworked by the microbial community which is in the sediments. In this experiment, a sand tank was filmed with Florida marine sand sample, and the interface layer was covered with oxygen sensor foils. You can also see in here the small tar balls. Now, in this experiment, there was a constant flow from the right side towards the left side of the experiment. And this flow also caused a ripple structure of the sediment surface. And if we now take a look at the oxygen profile over the sediment-water interface, you see that it's not a very steep homogeneous oxygen gradient over that interface, and the oxygen maps look totally different.
I can interrupt with a question here because it fits very well. Can you also see the interface, the water-sediment interface on the pictures?
Yes, the oxygen sensor foils or also the pH and CO2 sensor foils are semi-transparent, and you can see the interface structures in the recorded images. You cannot see them in the reference sensor response, but you will see it in the raw sensor response.
Great.
Okay, so now back to what we see in the oxygenation image of, at the water-sediment interface. Here you see the ripple structure of the sediment-water interface, and you see some ripple crests in here and also some ripple throats. And under the ripple crests, poorly oxygenated pore water from deeper parts in the sediment get pushed upwards, causing these large hypoxic areas. Whereas in the ripple throats, you see that highly oxygenated pore water is pushed inside the sediment and causing zones with higher oxygenation levels. So you see that the oxygen within the sediments is really not uniform. And with two-dimensional imaging, you can get an overview on what the oxygen profile looks like. So now let's head on to our tar balls, which should be reworked by the microbia. This black circle in here represents the area where the tar ball is located. And at a very early stage of the experiment, after some days, you already see that there is a microbial community consuming up the released hydrocarbons by the tar balls and also respiring oxygen while they are doing it. If you take a look at the later stage, for example at day 6, you see that there's even a higher oxygen depletion around the tar ball. And if you go on further, you can see that there's a really huge amount of microbial community around the tar balls, reworking the hydrocarbons that are released and consuming up the oxygen, causing an oxygen depleted zone. So now let's head on from sediments to other interfaces. Interfaces cannot just be sediment-water interfaces, but also, for example, water and air interfaces, and also intermediate interfaces such as the capillary fringe. The capillary fringe is the intermediate zone between groundwater saturated sediment and an aerated saturated zone. And in this immediate zone, gas exchange processes can take place. In this experiment, the gas exchange from oxygen and CO2 in that sample was recorded over time. There was an oxygen-rich water phase, oxygen-poor water phase, but CO2-rich water phase, and the atmosphere had a high oxygen content but a low CO2 content. And now the gas exchange properties within the capillary fringe were studied. You can see that in here in this video. So we will see the indiffusion of oxygen into the capillary fringe and the water phase, and an outgassing of CO2 over time. You can also see that this line in here represents the capillary fringe level, and the gas exchange is faster than the drop of the capillary fringe.
Well, while we see that video, there is another question concerning time. So, how long can you use the sensor?
Actually, this depends a little bit on the overall exposure of light to the sensor. We recommend to cover the foil whenever it is possible. For example, if you just take images every day, you just cover it with a black towel, and then it can be used for a very long time.
There's another question. Is it necessary to perform the measurements in the dark?
Actually, it's not totally necessary to perform the measurements in the dark. It would be preferable, but the VisiSens setup consists of an adapter tube which shields ambient light from the sensor signals. Okay, now let's head on. My next capillary fringe example is an aerobic biodegradation process of an organic model compound by the bacteria Pseudomonas fluorescens. Here on the left side, you can see the experimental setup. This was a large-scale sand tank. Large-scale, in this case, means a length of 80 centimeters and a height of 30 centimeters. And there was a groundwater inlet, which included the organic model compound, and a groundwater outlet. So there's a constant water flow from the left side towards the right side. This water-saturated zone is then covered by a capillary fringe zone and an air-saturated zone on top. The sand was inoculated with the bacteria so that it's evenly distributed, and then oxygen, pH, and CO2 was imaged over the whole surface. In this experiment, it was conducted with sensor strips positioned one beneath each other, and the cameras were moved from one position to the next position with a linear positioning system. They scan up the whole region, and after they've done that, they start all over again, and so you get a time series measurement in a large-scale view. I prepared a short video on that, where you can see on how the experimental setup looked in detail. So here you have the linear positioning slider, the sand tank, and the device. And here the cameras are moving from one position to the next one. The software is automatically recording the images while they have reached their position. And now let's take a look at the results of the experiment. I will just shortly press on stop in here to get a better view on that. So you see that within the capillary fringe is really a highly biologically active zone. And within that capillary fringe zone, the bacteria consumes up the organic model compound. And while it's doing that, oxygen is depleted because oxygen is respired by the bacteria. So if you monitor that over a time scale of 160 hours, you can see on how the oxygen depleted zone develops. So now let's take a closer look at the pH value. In 1 second. Now, in the pH imaging view, you can see that there's hardly anything happening. There's just some slight pH changes, a little bit due to noise, but in the active zone, the pH is pretty stable. So now if you take a closer look at the CO2 concentration over time and two-dimensionally recorded, we can see that at the inlet, above the inlet in the capillary fringe zone, CO2 is produced largely by the bacteria while consuming up the model compound. So now we have seen some experiments on sediments and also on interfaces. I want to now head on to my third topic of today, to imaging in biofilms. Before I start with my biofilm experiments, I want to show you a very easy model experiment. In this model experiment, E. coli colonies were grown onto an agar plate and then were covered with an oxygen sensor foil, and the oxygen respiration of these bacterial colonies and also in the media was investigated over time. The backside of the oxygen sensor foil is impermeable to oxygen, so what we get with overlaying is a small reservoir of oxygen, and this is then consumed up by the bacterial colonies. And here in that image you can see that you can clearly see the oxygen profiles or the oxygen values over your single colonies. And if you take a look at the time video of that, you can see that the oxygen is consumed up pretty fast above the colonies, and when these zones get oxygen depleted, the oxygen from the media starts to diffuse towards these areas and is also consumed up. So at the end, after 20 minutes total time, you see that all the oxygen is respired up. So with imaging, you can select a region of interest over every single point in here in your recorded image. You can, for example, sit on different colonies and get the respiration profiles of the different colonies. You can also sit on points where no colonies are and get the media oxygenation drop down.
Rob, there's a question concerning this application. How is it about sterilization of the sensor foils with the E. coli measurement?
The oxygen sensor foils may be sterilized with 70% of ethanol, or you can also use ethylene oxide or gamma irradiation. In most cases, 1 minute of plasma treatment is also feasible.
And there is a question about the calibration. Is the system like pre-calibrated?
Actually, the system is not pre-calibrated, but for example, with the oxygen sensors, you just need to do a 2-point calibration and then you can get your oxygen values.
Okay.
Okay. Now let's head on to the biofilm experiments. In my first example, the experiment was on monitoring a membrane fouling simulated during the biofilm cultivation. In this image, you can see that there's a mesh-like structure in an aqueous phase, and on these mesh-like structure, the biofilm grows over time. So if you take a look at the mesh structure itself, you see on day 1 there's hardly any biofilm on the mesh structure. On day 7 you can see distinct biofilm growth, and on day 14 you can see areas with large volumes of biofilm. These images were also recorded using the VisiSens device, and they were the raw sensor responses recorded with a backlight illumination to see the underlying structures. So if you now take a closer look at the respiration profiles and respiration maps of these biofilms, you can see on day 1, where there's hardly any biofilm, the oxygen profile is pretty uniform and we have a high oxygenation status. On day 7, some points of the biofilm starting to respire up the oxygen and consume up the oxygen, and this can be seen in here in these images. And on day 14, where we have large volumes of biofilm, you can see that we have large zones totally depleted from oxygen, but also zones where the oxygen respiration was weaker. And what you also can see in here, if you compare the raw sensor response with the oxygenation image, you can see that even if there are large volumes of biofilm, that doesn't necessarily have to be a huge oxygenation drop. So the biofilm can have big volumes, but the respiration rate can be different. So with oxygen imaging, you can define the respiration rate. And you can do that, for example, by choosing an arbitrary ROI and sitting on a specific position and getting the oxygenation drop profile. My last experiment for today that I want to present to you is also a biofilm experiment, and this is on the assessment of oxygen depletion and biofilm structure grown in moving bed bioreactor carriers. Moving bed bioreactor carriers are these kind of plates, and on these plates there's again a mesh structure, and within these meshes the biofilm is grown. These moving bed bioreactors were normally placed in recirculation pumps, and industrial wastewater gets in there, and the biofilm consumes up the organic compounds that are in the wastewater and thereby cleans up the water. And now we wanted to see on how the impact of oxygen or the oxygen respiration of the biofilm is dependent on each single mesh in here. You can already see from the microscopic image that there are some regions where there's less biofilm grown and also some regions where there's a large content of biofilm. So now if you take a look at the time series video, you can see that at areas with large biofilm volume, there's mostly a very high consumption rate of oxygen, but there are also these kinds of negative hotspots where the oxygen still stays high and is then just consumed up by time.
May I ask here, one question came up during the last application example. Is the foil in direct contact with the biofilm?
Yes, you just place it on top, or you can put it below your biofilm. And then you get a direct contact, and then you can start imaging and see the respiration of your biofilm.
Okay.
Okay. So I hope I showed you with these experiments that there's a lot of things you can do with imaging and a lot of information that you can gain. But if you think that something was missing from the systems, I can tell you that we also have customized solutions. If you want to get more information on that, you can just contact me via email. For example, we have systems that can image up to DNA 4 size. We also have systems that can image in a very micro scale, but also systems that can measure all 3 analytes or can be moved with the linear positioning slider. So I hope I showed you that you can really have a huge area of applications possible with imaging of oxygen, pH, and CO2. And maybe we can talk a little bit more on your applications. Please feel free to ask questions right now, or you can also contact me via email afterwards. And at the end, I want to tell you that we are at the AGU fall meeting next week in San Francisco. So if you're also there, you can just stop by at our booth and you can see how the systems work in real life. Our booth number is 1204.
Okay, great. I have actually another question here. This is about I guess that's a user of our systems like the Firebox. So is it possible to use other presets, fiber optic transmitters for the VisiSens foils?
Actually, the readout principle of these two devices is pretty different. The Firebox device reads out luminescent lifetime, and we read out referenced luminescent intensity signals. So we are working with intensity-based ratiometric sensor foils, and the Firebox is based on lifetime sensors. Okay. So actually, it's not possible.
Okay, then there's another question. How is it possible to evaluate the system? I think this is partially already answered by your chart here. Just contact us, and then I guess we can arrange a system at the user.
Yes.
And yeah, I think again, thank you very much, Robert, for your presentation today. And I think yeah, there will be continued communication. Just send emails, either Robert or info@presence.de, and we hope that you enjoyed the webinar. Thank you very much, and have a great day.
Thank you. Goodbye.